everyone, and welcome to today's webinar, Untangling Automated Proteomics. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit www.beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box in the lower left corner and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I do also want to mention that there is a survey at the end of this webinar. If you could please take a moment and submit your responses when prompted to do so, that would be greatly appreciated. I'd like to now welcome our speaker, Dr. Sudha Savant, application scientist with Beckman Coulter Life Sciences. Dr. Savant, you may now begin your presentation.
Thank you, Cassie. Good morning, afternoon, evening to all of you. Today, I'm going to present to you on how automation can play a significant role in simplifying workflows in day-to-day proteomics. This is important as these solutions enable research in various areas of oncology, epigenetics, genomics, molecular biology, and biochemistry. At Beckman Coulter, our vision is to accelerate answers, and our mission is to empower researchers like you with tools to answer important scientific questions. So who are we? We are experts in fluid transferology. Saying so, let me walk you through our automation portfolio. Between the Biomech I series and the echo-acoustic liquid handlers, we can transfer liquids in the range of nanoliters to milliliter scale. The Biomech i5 workstations are useful for low to mid throughput workflows, and the increasing arrow shown on the deck represents growth with increasing throughput. The Biomech i7 has larger deck space, and the workstations have different instruments integrated to it. For our acoustic liquid handlers, we have the 525 series, which transfers 25 nanoliter droplets, and the 600 series, which transfers 2.5 nanoliter droplets. We also provide the Echo and Biomech consumables. So what is proteomics? Proteomics is the study of the proteome. It investigates how different proteins interact with each other and the roles they play within the organism. Each species has its own unique proteome. Unlike the genome, the composition of the proteome is in constant state of flux over time and throughout the organism, making it really important for research. Proteomic research provides a global view of the processes underlying healthy and diseased cellular processes at the protein level. Proteomics is applicable largely in the drug and biomarker discovery and is used in the fields of personalized medicine and systems biology. So why is it relevant and why is it used? So in the pharmaceutical industry, it is used for the production of antibiotics, monoclonal antibodies, and recombinant proteins. Other applications include in the food and beverage industry, in agriculture, and household care products. The proteomics has three main types: expression proteomics, functional proteomics, and structural proteomics. The other proteomics included are post-translational modifications, protein-protein interactions, and proteome mining. Protein expression profiling is an area in which proteins that are normally expressed in particular cell type, tissue, or organism as a whole, or proteins that are differentially expressed are profiled. It's commonly used in medicinal microbiology, signal transduction, and disease mechanism. Structural studies yield important insights into protein function, the druggability of protein targets for drug discovery and drug design. Functional genomics is focused on identifying the biological functions of specific individual proteins, classes of proteins, or whole protein interaction networks. Post-translational modifications are useful to study glycoxylation, phosphorylation, and proteolysis. Protein-protein interactions investigate how proteins interact with each other. Proteome mining is useful in the fields of drug discovery, target validation, and differential display. For each of these workflows, the downstream data collection is different. It can be crystallography, binding studies, use of capillary electrophoresis, or mass spectrometry. The sample preps for each is labor intensive and with high throughput requires more hands-on time. And there is no standard technique for sample prep in proteomics. Proteomic sample prep is tedious, and the reliability of the data analysis is largely dependent on quality of the prepared samples. Hence, automated solutions can help make proteomic workflows as pain-free as possible. Automated solutions generate reproducible samples with consistent quality and yields. They minimize user hands-on time, error, and bias, deliver value at the sample level by minimizing cost to sample, and increases overall efficiency and productivity. So let's meet the Biomech I-Series automated workstation. This is the workhorse for our proteomics workflows. The Biomech I-Series is extensively used for the mid to high volume sample preps for proteomics. It is simple, so you can focus on the science. It is efficient to deliver productivity. The Biomech i7 has been commonly used in the proteomic workflows that I will be showing you later on in this presentation. It's because it has a large deck space, so you can process 96 samples at a time. We have methods that are adapted to the Biomech i5, but these may have lower sample throughputs or maybe more longer times for processing. The i5 comes with one pod, while the i7 has two pods. So we have the SPAN8 pod. So the i5 can come either with a SPAN8 pod or a multi-channel pod, and the multi-channel can come as 96 channel head or a 384 channel head. The i7 has two pods, so you can either get two pods of multi-channel, two pods of SPAN8, or a dual where you have one SPAN8 and one multi-channel, which is commonly used in a lot of the workflows. The multi-channel head, that is the one with 96 channels, also comes as a 300 microliter capacity head or a 1200 capacity head. Biomech is ideal for proteomics as it's adaptable. It can be easily integrated with pressure alps, deck washers, heating and cooling devices, and incubators on deck. This is important as integrations provide a walkaway solution. The Biomech software is intuitive and icon-based, which makes it easy to set up methods for new users. And it also provides control over complex tip movements for the professionals. The Biomech software has a graphic user interface to write protocols and visualize how the workflow will progress. This simple yet sophisticated nature of the Biomech software makes it easy to implement amongst users of varying level of automation experience. In addition, Beckman Coulter provides software training for users, enabling a smooth transition from manual to automation. So here is a list of common proteomics integrations. Integrations are important, as I mentioned earlier, because they provide a walkaway solution. And we have an entire team at Beckman Coulter, known as the Access Solution Group, which works on integrating various third-party devices on the Biomech. Some of the common integrations in proteomics involve positive pressure ALP, plate readers, washers, centrifuges, and sonicators. We also have the SAMI, which is a planning scheduler, which is used to write methods that run on Biomech instrument. It plans all the individual steps required to execute a particular action schedules all actions to create the shortest and the most efficient method execution, and it also monitors the method as it executes. SAMIX treats all the sample the same way and runs consistently each time in the method when the method is run. This enhances the repeatability and decreases technical variations. So keeping this in mind, I would like to now walk you through some of the proteomic workflows that we have automated. So I have just made a general list. Proteomics has wide variety of overlapping techniques for to ask the research questions. I have categorized the automated workflows based on general use. These may overlap at different points. Like example, mass spec can be used for functional proteomics or structural. This is just a very general classification for the ease of the presentation. For all these different workflows, it is important that the liquid handlers are able to integrate to different third-party devices or just different devices. So the first on the list is the Bradford assay. Bradford assay is a common total protein assay similar to the BCA or another quantitative colorimetric assay. Although simple, the repetitive pipetting steps in the Bradford assay make it cumbersome to carry out manually, especially when the throughput is high. Based on the desired throughput and walkaway, Bradford method can be implemented on the Biomech i5 Span 8 system, the Biomech i5 multi-channel, and the Biomech i7 hybrid, along with the SpectraMax i3 plate reader from Molecular Devices. This slide provides detail into the quality of the output on the Biomech I-series instruments. So in this app note, we set up the Bradford assay on all three workstations, and the R-square was more than 0.99 for all three systems, which is the i5 Span 8, the i5 multi-channel, as well as the dual i7 hybrid. Please note the consistency between the samples in the low concentration ranges, with the CV below 2% for all these samples. This shows that the Biomech can provide consistent results in an analytical assay. We also have an example of the ELISA on the Biomech I-series workstation. So enzyme-linked immunosorbent assay, or ELISA, is a plate-based assay technique that is often utilized in expression studies to verify the abundance and specificity of the binding of an enzyme, protein, antibody, or hormones relative to a control. This immunological-based assay comes in 4 unique types: direct ELISA, indirect ELISA, sandwich, and competitive ELISA. The sandwich and competitive ELISA are the most utilized in the clinical research field. The ELISA assay setup is pipetting heavy with tedious repetitive washing steps. Precise pipetting is crucial for a successful assay, and while doing it manually, even shifting focus for a few seconds can result in errors. Missing even one reagent can lead to weak signal or no signal. So automating ELISA is really helpful to minimize user error and contamination. It can also help reduce user hands-on time. With automation, you can also trace your samples with the data acquisition reporting tool system that we provided from Beckman. What you're looking at is data of utilizing the Biomech i5 for a direct ELISA technique. The data yielded on the Biomech for triplicate values has 30% lower than 5. This reflects the ability of the Biomech i5 to uniformly process samples across the plate. This figure shows the linearity of 0.991 that illustrates excellent performance of the standard curve dilutions. Some other examples for IgG measurement automated on the Biomech are the Velita titer. We also have miniaturized alpha LISA screening by tenfold using the Echo liquid handlers. Next, I want to discuss sample prep for mass spectrophotometry. So for Demonstrating sample prep for mass spectrum photometry, we have collaborated with Cedar Sinai, Dr. Jennifer Van Eyck, who is an international leader in the area of clinical proteomics, and her lab has focused on developing technical pipelines for de novo discovery and large-scale quantitative mass spec methods. Her lab is well known for the extreme technical quality of the data generated, the rigorous quality control with tight percent series, while applying these to key clinical questions. Their aim of the lab is to maximize throughput and reproducibility to move targeted and robust discovery methods into large population. So this is a deck layout of the Biomech i7 that is at Cedar Sinai. It's commonly been used for protein digestion and desalting for the preparation of samples for mass spec analysis for biomarker discovery. We have successfully collaborated and implemented these two methods, which resulted in a 7-fold reduction in time and a 4-fold improvement in percent of correlation of covariation. This is the deck layout. The LF stands for labware feeders, which is basically a space to keep plates and tip boxes so as to free up your deck space. PL stands for tip loading. So these are the positions where you have the tip boxes present, where multi-channel head can load the tips from these boxes. TR are the trash positions. The P positions are the static out positions for your plates, reservoirs, reagents. The Peltier 1 is a heating or cooling position. In HECO is the incubator that is used for, particularly in this case, used for digestion. The orbital is for shaking. And PPA is the positive pressure apparatus. This is from Amplius. And in case of Cedars, we use it for their automated desalting. So the PPA is a solid phase extractor, and it is equipped with a clamping pressure of 0 to 3000 psi and a well pressure of 0 to 1500 psi. It has a clamping arm that has 96 holes to deliver same pressure to each well and a moving tray with a vacuum drain. The Biomech places the adapter with the collection plate and the plate with filter columns on the PPA. The PPA is great to even do partial plate preparation as the pressure is evenly distributed. PPA can also be used for solid phase extraction, buffer exchange, sterile filtration, and sample concentration. So let me get into the details of the experiment that we did to show how automation can help with robust sample prep. The aim of the experiment was to identify biomarkers from cerebrospinal fluid and naive plasma. So both the biofluid digestions were done on the Biomech i7 hybrid station using a 96-well plate. For digestion quality control, each plate combined 4 quality control reference samples along with patient samples. These quality control reference samples were placed in 4 wells of a 96-well plate. So that is to designate each quadrant of a 96-well plate. The cerebrospinal fluid and the naive plasma proteins were denatured, reduced, and alkalized, followed by trypsin digestion on the Biomech i7. So there were 25 plates for cerebrospinal fluid and 13 plates for naive plasma. For the cerebrospinal fluid, there was no desalting performed. It was analyzed on the EvoSep Explorers 480 system. For the naive plasma, after the digestion, the desalting was also done on the i7, and this was then analyzed on the SciX TripleTOF 6600 system. This is the percent CV for the interday and the intraday for the cerebrospinal fluid and knife plasma, protein, and peptides. As you can see, it is less than 16%, which shows robust sample prep and reproducibility on the Biomech I7. These graphs are showing the spread of the proteins and peptides from each of the fluids, either the plasma or the cerebrospinal fluid, um, from the four quadrants from multiple plates. Another exciting workflow that we have collaborated with Cedars-Sinai is on the cis-kappa workflow, which stands for Stable Isotope Standards and Capture by Antipeptide Antibodies. This workflow uses antibodies which are raised against specific peptides for specific proteins and then coupled to magnetic beads. Plasma samples are added to a denaturing plate to denature the proteins. Then the proteins are reduced, alkylated, and digested. Next, the antibody beads and heavy peptides are added to allow for coupling of the target peptides. This is followed by bead washing and elution. And then the elute is subjected to mass spec analysis. So again, as you can see, this is a manually extensive process with multiple washes, incubations, and wait times. And so automation can really make it easy to do a 96 sample prep or even a 384 sample prep simultaneously. This workflow has previously also been automated on the FXP and NXP instruments and is extensively validated as a reproducible workflow. So we did this on the I series, which is the I7, on the same deck layout as I showed you previously for the protein, for the mass spec. So to demonstrate the capabilities of the cis-kappa method, what we did was that there were 2 columns of whole human plasma were processed, so 16 samples in total. To assess the reproducibility, the eluates from one column, so that is 8 replicates, were combined, and this was to assess the imprecision resulting from mass spec measurement. The remaining 8 replicates were measured individually to determine overall coefficient of variation. To calculate the imprecision executed on the Biomech I7 platform, the sum of the squared models was employed, which is this coefficient of variation of the I7 was the coefficient of variation of total workflow minus the one that was coming from the EcoMS system. And as you can see, for the Biomech, the CV percentage is less than 8. And this indicates that the Biomech, the sample prep on the Biomech is highly reproducible. We have, so far we have seen examples of the mass spec prep, and so we will move forward to look at sample preps from capillary electrophoresis that can be used for functional proteomics. Protein therapeutics is a rapidly growing industry for treating serious illnesses like cancer, autoimmune disease, and capillary electrophoresis has a strong presence in all stages of a therapeutic protein life cycle. So this slide pretty much says it all. Every stage from cloning to transfection to process development, purification, and quality control. Capillary electrophoresis plays a very important role in characterization and validation of many of these stages. It can either be in glycosylation analysis or in the purity and heterogeneity of the charge on the monoclonal antibodies. Capillary electrophoresis is of interest since it provides high resolution and efficiency of separation. The BioPhase 8800 system from SciEx can achieve 8 times faster charge heterogeneity analysis as compared to a single channel CE since it has 8 capillary electrophoresis channels. So the SciX kits for the Biophase 8800 system automated so far are the Biophase fast glycan labeling and analysis kit, the Biophase capillary electrophoresis SDS protein analysis kit, and the capillary isoelectric focusing kit. So I'm going to first begin with the details of the fast glycan labeling and analysis method. This was carried out on the Biomech i5 multi-channel liquid handler with the 300 microliter head capacity. So for the fast glycan, it involves mainly the denaturing of the protein, then the release of the N-glycans, the labeling of the N-glycans, and then cleaning of the X to remove the excess dye, and then ultimately the elution. So once again, this sample prep is tedious and has a lot of wash steps and pipetting, and therefore automating this workflow makes it easy for the user and can remove and can minimize user errors. The total preparation time for a 96 well plate was approximately 2 hours and 37 minutes. This figure shows overlay of multiple injections showing consistency between sample preps and analysis. The robustness of the automated sample prep was assessed by evaluating the intra and the intercapillary consistency for the corrected peak area percentage or the CPA percentage and the relative migration time of the glucose units, which is the same as the relative migration time of the glycan.
It looks like we may be having a slight connection issue. Sudha, if you want to go back just a little bit, you were paused for a while, just so you're aware.
Can you hear me now? Where was I paused at? Do you know which slide?
Right at the very end. So if you just want to go over the last part, and we can hear you again.
This fast glycan part?
Yes.
Okay. All right. Okay, I'll just redo the fast glycan because I'm not sure how much was lost. So for the SciX kits for the capillary electrophoresis or the Biophase 8800, we use the Biomech i5 multi-channel liquid handler with the 300 microliter capacity head. The fast glycan labeling method involves denaturing of the protein, then release of the N-glycans, followed by the labeling of the glycans, removal of the excess dye, and then elution. Each of these steps involves a lot of manual pipetting and a lot of incubation time as well as washes. The total preparation time for the 96 well plate was 2 hours and 37 minutes on the Biomech. This figure shows overlay of multiple injections showing the consistency between samples. The robustness of the automated sample prep was assessed by evaluating the intra and the intercapillary consistency of the corrected peak area percentage, the CPA, and the relative migration time of glucose units, which is the same as the relative migration time of the glycan species. And the values for the CPA and the relative migration time are given, like it's an Excel file that is you can get from the Biophase software, and then you can do the analysis on it. So the data was analyzed manually, and as you can see, between all the 96 samples, the relative migration times look consistent, and this shows that the sample prep was reproducible and consistent. So now let's look at the capillary electrophoresis STS protein analysis kit. So one kit can prep for up to 200 samples. So for the biophase 8800, it involves, so it has 4 trays that you can put in. So that is the sample inlet tray, the reagent inlet tray, reagent outlet tray, and sample outlet tray. For the fast glycan, we have shown the preparation of the sample inlet tray only. For the SDS, because the sample prep is relatively easier and less steps, we have within one method all the 4 trays that can be prepped. And the way the method is set, the user can choose to do only one prep at a time, or you can do all the 4 trays prepped in one go. So the methodology involves heat denaturation of a specified concentration of protein in the presence of SDS. Once denatured, the sample is separated by size in the capillary and selected based on size. So this table provides the different times for each of the individual tray preparations. So the total time for the reagent inlet outlet, the sample inlet outlet, can be reduced if you use a 1200 multi-channel head. The times here are higher because we had a 300. So at a time, we could only pipette 300 up to 300 microliters. And the volumes for the reagent outlet tray, sample outlet tray, reagent inlet tray are more than 300 microliters. So we had to do multiple trips. This is the data for the SDS demonstrated method. On the left is the electrophoresis for 96 reduced monoclonal antibodies. Peak 1 is the 10 kilodalton marker, which is used as an internal standard. Peak 2 is the light chain. Peak 3, which cannot be seen here, but maybe in the single run, little blown up, is the glycoxylated, is a non-glycoxylated heavy chain, and the peak 4 is the heavy chain. The different colors, A to H, denote the 8 capillary numbers, with the first run starting from the top. And this is a single electrogram of a run, showing again the 4 peaks. To determine the inter and intra-capillary reproducibility, we analyzed once again the CPA and the relative migration time of the species of the reduced IgG, and the ratio between the heavy chain and the light chain, and the resolution between the non-glycoxylated heavy chain and the heavy chain. So this resolution really speaks volume for the way the sample was prepped, because if you do not have good mixing and sample prep, then there will be no resolution between these 2 peaks. And the resolution for the automated prep between the non-glycoxylated heavy chain and the heavy chain was better than 0.87%. This table once again shows consistent migration times and CPAs for all the 4 peaks, and less than 3% relative standard deviation, which indicates that the assay is specific, reproducible, and robust for individual samples. This method has also been demonstrated on the SPAN8 I5 system, for which we have an app note. Okay. Last, I want to discuss the CIEF kit. So this method is very similar to the SDS, with once again the sample inlet, sample outlet, reagent inlet, and outlet tray preparation. These are the times specified for each tray, and the total runtime for this method is 2 hours 20 minutes, which once again can be reduced if you use a 1200 microliter multi-channel head. So the electrogram is showing absorbance and the isoelectric point value for the main isoform of the IgG. So it's basic, the main isoform, and acidic. These 3 peaks are the internal standards that we use, which was proteins with isoelectric value at 4.1, 5.5, and then at 10. This is this is an electrogram from a single run. So just to show the individual peaks, I know it's harder to see it here. So this is once again the detection time for the main isoform as well as the standards, and the CPA, which shows the robustness of the assay. You can see how tight the data is between each of for all of these groups. This table once again shows how consistent. So let me just explain a little. So this is the sample columns. So what we are looking at is instead of the capillary, we are looking at the different columns. So for same capillary, and you can see that the values are pretty consistent for the calculated isoelectric point as well as the detection time. And we have great relative standard. We have very low relative standard deviation, which once again indicates robust automated sample prep. Okay. Another important assay in the functional proteomics that is commonly used is the luciferase reporter assay. The dual luciferase reporter setup demonstrated here used a firefly luciferase reporter to detect NF-kappa B signaling in response to tumor necrosis factor alpha, TNF alpha. The control luciferase with constant expression used was NanoLuc from Promega. So the Biomech i7 system that we use for this assay was equipped with a HEPA filtration unit, and this is to prevent the contamination of the cell cultures while they are on the Biomech deck. 48 hours later, the automated method removed the media and added the TNF alpha to the appropriate wells. After a few hours, the luciferase reagents were added again on the Biomech i7 station, and the plates were then read automatically using the integrated SpectraMax i3 plate reader. In total, the only user intervention that was required, other than the setup, was moving the plates manually from the Biomech to the incubator. And this can also be eliminated if the system is integrated with the automated cell incubator, such as a Cytomat. This will allow for end-to-end performance, and so the plates will never have to leave the Biomech environment. So we compared the manual results with our automated method for a 96 well as well as a 384 well assay. And you can see the automated method, if not better, performed well. It was actually better as compared to the manual. For the 384, once again, the difference is really strong on the automation compared to the manual. The 384 well was done to show that we can do miniaturization, and this which allows to increase throughput and reduce reagent costs by almost 4 times. Okay. So I have an example of the ECHO 600 and the 500 series to show how the ECHO liquid handler can be used in the field of structural proteomics for crystallography. The ECHO liquid handler enables protein crystal formation with sitting drop volumes as low as 50 nanoliters. Nanoliter volumes of protein and crystallography reagents can be transferred into sitting drop wells of crystallography plates. Precise and accurate drop placement eliminates the cross-contamination and ensures drop-on-drop placement for blending protein and crystallography solutions. Precise transfer of reagents ensures repeatable crystallization trials. With the dynamic fluid analysis, only a single fluid class setting is needed to transfer all the crystallography sparse matrix reagents, eliminating the need for multiple calibrations or repeated optimization of fluid class settings. So in what you're seeing here is an experiment. So there were three experiments done. The first was to test the drop transfers, which the data is not shown on the screen. The second was the different amounts of protein and drop volumes, which is seen in this figure one. So what the aim was to see if when you miniaturize the protein or the drop volumes of the crystal reagents, are we getting the same shaped crystals or not? So the first column is the 50 nanoliter drop. The second is the 100 nanoliter drop, and then is the 1 microliter manually performed experiment. And all three gave the same shape of the crystal to say that miniaturizing, so this shows that miniaturizing the experiment did not shift the crystal structure or bring a change in that. The same experiment was then done in triplicates. So for the echo, we had 144 points, and for manual, we had 288 points. And what it's saying is that what this graph shows is that there is consistent crystallization percentage yield, even with miniaturization, as compared to manual. So with this, I would so these are all the automated preps that we have. We have many more, and you can go on to our website to look at the app notes in detail. I hope that I have convinced you all about the importance of automation for proteomic workflows to acquire consistent, reproducible data. I want to thank you all for listening, and I'm happy to answer any questions that you may have.
Thank you, Dr. Savant, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the lower left corner of your screen, and we'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, the ELISA workflow as described looks to be very tip heavy. Is there an alternative option?
That's a very good question. Yes, so we have done this previously, and what we can do is integrate plate washers with the biomech, and this will significantly reduce the tip usage for in-between washes.
Okay, great, thank you. Our next question here says, when automating the CE-SDS protein analysis kit, you mentioned 2 user interaction points. Can you elaborate on which points in the workflow a user must intervene and why?
Yeah, that's a great question and catch. So the first user intervention is actually when you set up the deck for the experiment. So that's always going to be there for any experiment. For the CESDS protocol, while we were demonstrating, when we placed the plate on the heater for the denaturation of the protein, we did cover it with we had to pause the biomech to add a foil or a plastic covering so as to prevent evaporation. We at Beckman we do have microprime lids which help with evaporation, but we just don't have enough time to test it out in this case, but that is something that can easily be done.
Okay, great, thank you. And it looks like we have just one more question here. That question says, you also mentioned mixing well. Is that well, and how could you tell when developing the method that it was sufficient? Or what is well?
Mixing well is basically, especially when you have small microliters that you are mixing the fluid. So it's ensuring that that the reagent is pipetted and it makes a homogeneous solution. So when we are developing methods, we generally use colored fluids, colored water, or colored reagents. And so when we do tip mixing, you can visually see if the mixing has been done, has been is perform the liquid handler is performing the mixing well or no. Additionally, I mean, the data is always something that can be that can will speak for itself.
Wonderful. Thank you again, Dr. Savant. Do you have any final comments for our audience today?
No, just thank you once again for listening.
Great. Thank you again, Dr. Savant, for your time today and your important research. We would also like to
Thank you.
thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. I want to remind everyone that there will be a survey that pops up at the end of this webinar. If you could please take a moment to submit your responses before you leave. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Bye. Thank you, Cassie.